Diploid eukaryotic cells must reduce their ploidy by 1/2 in order to make haploid gametes to continue the next generation. A diploid cell undergoes 2 cell divisions to make haploid gametes. When the haploid gametes recombine, either by fertilization of an egg or a sperm, or by syngamy, where 2 similar haploid cells fuse together, the zygote is diploid. The reduction in chromosome number to make the gametes by cell division is called meiosis. A single diploid cell undergoes 2 cell divisions to make 4 haploid gametes. Interphase in meiosis is just like in mitosis. The cell increases in volume and produces all the molecules needed for the cell to survive and adapt to its environment. When the cell is large, the DNA duplicates just like in mitosis, and is very similar to mitosis, except now the cell is preparing for meiosis instead of mitosis. Meiosis is divided into 2 stages, meiosis 1 and meiosis 2. Each stage in meiosis is divided into 4 separate phases like in mitosis. The first phase is prophase 1, and like in mitosis, the chromosomes condense and the centromeres become visible. The first major difference with mitosis now occurs, the chiasmata form. Chiasma is the singular. During prophase 1, the homologous pairs of chromosomes line up next to each other. So the red jacks line up with the black jacks, the red 3s line up with the black 3s. And the 2 sister chromatids from each chromosome will exchange pieces, so now all 4 chromatids are physically linked through the chiasma. The 2 red chromatids are linked at the centromere, 2 black chromatids are linked at the centromere, and because of the crossover, the 2 homologous pairs are linked to each other through the chiasma. So now all 4 chromatids are linked together in a single physical unit. It's completely random where the chiasma form between the chromatids. In every meiosis, every crossover is random. This is the first step in creating genetic diversity in meiosis. And then, like in mitosis, the nuclear envelope dissolves in some of the species of algae, and then the spindle forms. In metaphase 1, the homologous pairs of chromosomes line up at the chiasmata, not the centromeres. It is completely random how the homologous pairs orient themselves with respect to the poles. In this case, the black jacks, black 9s, and red 3s are on the left side. In another cell, it might be all the red homologous pairs are on the left. How one homologous pair orients itself has no effect on how any other pair orients itself. This is the second step in generating genetic diversity in meiosis. In anaphase 1, it is the centromeres of each homologous pair that are pulled towards the poles. They do not disintegrate at this point like they would in mitosis. In this process, the chiasmata break. It is a 50-50 chance whether the chiasmata break and give you back the chromosomes you started with, or they swap pieces. If they swap pieces, it creates chromosomes that did not exist in the parent cell. This is the third step in creating genetic diversity in meiosis. When the centromeres are pulled towards each pole, the chiasmata will break. A break in the horizontal direction will give back the chromosomes that you started with. A break in the vertical direction will exchange pieces that were crossed over. This exchange of chromosome fragments will create new chromosomes that did not exist in the parent cell. At each anaphase 1, you can have one or the other. Chromosomes that are created by this mechanism are called recombinant chromosomes to distinguish that they came about by recombining the genetic material. In telophase 1, the chromosomes may or may not decondense, and the nuclear envelopes may or may not reform depending upon the species. The spindle will disassemble itself, and there is a complete set of chromosomes at each pole, but they are no longer identical sets. And as you can see with the 9s, some new chromosomes have been created when the chiasma broke in anaphase 1. Interkinesis is the period between meiosis 1 and meiosis 2. It is kind of like cytokinesis at the end of mitosis, except the cells do not continue into interphase. They continue into meiosis 2. At the end of meiosis 1, the daughter cells are not genetically identical to each other or to the parent cell that they came from. Meiosis 2 is the second cell division in meiosis. In prophase 2, the nuclear envelope dissolves in certain species, but not others, We will learn the differences later in the course. The spindle forms, and the chromosomes condense if they change back into chromatin, the loose yarn, at the end of meiosis I. In metaphase II, the condensed homologous chromosomes attach to the spindle along the equator by the centromeres, just like in mitosis. The only difference is that the chromosomes are now considered haploid because the red and black cards are in different cells. Anaphase II is just like anaphase in mitosis. The centromeres dissolve, and the chromatids are pulled to the opposite poles. Telophase II is the same as telophase in mitosis. The spindle dissolves, the chromosomes decondense into chromatin, and the nuclear envelopes reform. Cytokinesis is the same as in mitosis. The cytoplasm divides the two daughters from the end of meiosis I into 4 haploid cells. If the cells didn't divide in interkinesis, cytokinesis at this point creates 4 daughter haploid cells. Each daughter cell has a complete haploid genome, 1 3, 1 9, and 1 Jack. But they are different sets of chromosomes. None of the sets of 3s, 9s, and Jacks are the same in any cell. And now meiosis is complete. 1 diploid cell has given rise to 4 haploid daughter cells for reproduction. Unlike mitosis that duplicates the deck, or the chromosomes, meiosis does shuffle the deck by the random formation of the chiasma in prophase I, and then the random orientation of the homologous pairs in metaphase I, and finally the 50-50 chance of swapping pieces of chromosomes when the chiasmata break in anaphase I. At the end of meiosis, there are 4 haploid cells, each with a complete set of chromosomes. But the sets of chromosomes in each are not identical. As you probably guessed from the description of the stages of meiosis, not all species do sexual reproduction the same way. In fact, most species on Earth do not sexually reproduce the same manner animals do. In order to begin the discussion of reproductive life cycles, we need to learn some new vocabulary. In sexual reproduction, there are gametes. These are the haploid reproductive cells that are a result of meiosis and have a male or female version. Male and female are in quotes because the distinction between the sexes can be very difficult in many species. And in many cases, the sexes are distinguished by a plus or a minus. After the haploid gametes fuse, the resulting diploid cell is called a zygote. In asexual reproduction, many algal species produce spores. Spores are specialized cells that can give rise to a new individual without sex and can be either haploid or diploid. What distinguishes a spore from a zygote is that the spore can be dormant for long periods of time while waiting for favorable environmental conditions for germination. There are different types of spores. Zoospores are motile and use flagella for locomotion and lack a true cell wall. The planospores are non-motile, without flagella, and with a cell wall. They rely on passive transport by the currents of air or water that they are in. Autospores are non-motile spores produced in and have the same shape as the parent cell. Finally, there are cynobia. These are loose associations of single cells forming a sort of colony. Unlike an organism that is a single individual made of many cells, a synobium is an organism-like group of single cells. The reproductive life cycle you are most familiar with is the diploid life cycle. Haploid gametes are produced in meiosis. They fuse at fertilization to make a zygote. The zygote grows into an individual by mitosis, and eventually makes gametes by meiosis to start the cycle over again. In the diploid life cycle, the haploid phase is very short, and most of the organism's life is diploid. In the haploid life cycle, when the gametes fuse to create a zygote, the zygote does not undergo mitosis. It undergoes meiosis to create haploid spores. After germination, these spores grow into mature individuals by mitosis. These mature individuals are haploid, but either as male or female organisms. These haploid individuals will create gametes by mitosis. In the haploid life cycle, gametes are not created by meiosis, but by mitosis of the haploid parent cells. Organisms at this type of life cycle will spend the majority of the life cycle as haploids and only a very short time as diploids. Alternation of generations is just that. Half of the time the organism is haploid, half of the time it is diploid. Mature haploid individuals are called gametophytes, gamete-producing individuals. They produce gametes by mitosis. The gametes fuse and become a zygote that grows by mitosis into a mature diploid individual called a sporophyte, or spore-producing individual. The sporophytes produce spores by meiosis. These germinate and grow into gametophytes by mitosis after germination. In some species, the sporophytes and gametophytes look identical. In other species, the sporophytes and gametophytes look very different. And in some cases, the sporophyte and gametophyte are so different they were named as different species. The ability to sequence the genomes of organisms today has greatly helped to unravel the confusing state of affairs. As an example of this extreme state of affairs, in 1843 the species Porphyra umbilicalis was described. It is a large foliose red alga that is the source of the food dulse. In 1892, Conchycelis rosea was described. It is a microscopic red alga that lives on the surface of hard things like seashells. And this was the state of affairs in red algal taxonomy for a long time. The form of Porphyra that we see is the gametophyte form. But the sporophyte could never be found in nature. The sporophyte had been found, but had been named a different species. Conchycelis rosea is actually the sporophyte form of Porphyra umbilicalis. The sporophyte forms of Porphyra species are now called the Conchycelis stage. We will learn more about this life cycle arrangement in future lectures.